And we have that Galaxy platform. So my partner, that is another NGS analyst, will going to show us how we can perform the whole analysis on Galaxy platform. So he will tell us about Galaxy platform and he will guide all the features related to the Galaxy platform. Then we have the methodology. Look, we have different types of methodologies in this particular analysis. So we will focus on a particular method or particular pipeline that we are going to follow for the whole analysis. And we will going to highlight that also. Now we have some important steps in this whole training that required quality check by the fast QC. So we use fast QC and we will perform the quality check because it is very much important if we are dealing with the genomic data. So we need to perform the quality check. So we should eliminate out the useless information or useless sequences. Then we have the error read correction. That is another step that is essential for the whole analysis. Then we have that identification of bacterial species. We have selected bacterial species because bacteria has a limited amount of genome as compared to other species. Bacteria are prokaryotes and they have incomplete genetic information as compared to other organisms because other than bacteria, all the organisms are eukaryotes. They have the complete genetic information. So if we focus on the bacterial species, we can easily perform the analysis in quick time, and we can also highlight many features related to that particular genome of the bacterial species. Then we will do the antimicrobial resistance gene identification. So we will targeting the antimicrobial resistance gene because we need to understand the gene that are involved in the resistance, like which particular segment or which particular part of the genome is involved to make the bacteria a much stronger species. So we will focus on the identification of antimicrobial resistance gene, and then we will perform the pathogenicity and pathogenic gene identification. And in the later half of this particular training, we will also perform assembly and annotation. So after sequencing, we have assembly and annotation. These are two main or two key features in the whole analysis. So we will perform assembly with the help of multiple assemblers, like we usually use spades. There are some other assembler as well, like Velvet and other assembler also. But we will use spades for assembly, and then we perform the annotation and visualization. And we have different softwares to perform this annotation and visualization, and we have different results related to this. So we need to understand how we can analyze the result that is generated after the assembly and annotation, and what kind of graph we will obtain by the whole analysis. So we will focus on different graphs and different types of representation related to the results of whole analysis. Okay, so this is the main features. This is the main objective that we are going to perform, and we will discuss in detail about each and every single step listed over here. Okay, so now we have some other information related to the training and about the Galaxy platform. So we'll go into that particular part. Okay, we have another participant. So we will going to add that. Okay. Okay, this is the Galaxy platform. Okay, I'm just giving you the basic information to let you know that what we are going to do in the future and the upcoming training. And I just need to highlight some features that are listed over here on the left hand side of your screen. Like you can see, we have various tools. We have genomic file manipulation. We have different types of files that is generated in this particular segment. Then we have common genomic tool that are very much essential to manipulate the genomic data. And then we have the analysis. So we can perform different types of analysis, like we can do assembly, annotation, mapping, variant calling, chip sequencing, RNA sequencing, phenotype association, evolution. So we can perform different type of genomic analysis. So in this particular part, we will discuss all these particular things that are listed over here. When we discuss about Galaxy, we will let you know that how this particular platform work and what kind of tools are embedded in this particular platform. So Osama will tell you in detail about this particular platform. Here we have different type of analysis related to metagenomics. And I think some of you have the information related to metagenomic as well and microbiome as well. So we will just going to give you some basic information related to that kind of analysis. So you have to wait for that particular session. But here we have all these things associated with the Galaxy platform. Okay. Okay, this is some basic information related to NGS, and specifically we can talk about the Galaxy platform and different things related to this. So this is the official document from NIH, National Institute of Allergy and Infectious Diseases. So this particular institute provide the data for genomic sequencing or genomic data analysis. So we have some basic information listed over here that is essential for the training that we are going to perform. So if we move to the top part, here we have the information related to Galaxy platform. Okay. So if we see over here, we have the link, and then we have some filter that we can use for performing different type of analysis or for doing different steps. Okay. Makes analysis method available to the community and facilitate the reproducibility via creation of reusable workflow. It means that this particular platform is helpful for reproducing the result and modifying the result. If we need to add something or we have to perform another type of analysis, we can use the data that is just generated